Good morning, class. Today we are going to talk about cell culture. What is cell culture? When we take out a tissue from its normal position and culture it in laboratory, it is known as cell culture. Cell could be of animal origin or plant origin. The environment in which we culture is artificial environment which is created in the laboratory. In laboratory, we use a glass or a plastic vessel containing liquid or semi-solid support medium which supplies the nutrients for the culture of the cell. When the organs are removed and the cells are removed from the organs, the normal relationship is disturbed and the cells are cultured in the laboratory. It is a cell culture. Classes of cell culture. We can have primary cell cultures, we can have cell strains, and then finally we can have cell lines. Now what is the difference between all three? Primary culture. When the cells are removed surgically from an organism and placed into a suitable culture environment, they will start attaching to the media, then divide and then grow. Most of the primary cultures, they have a finite lifespan of approximately 5 to 10 divisions in vitro. Vitro means in laboratory conditions. Due to the limited lifespan, we cannot have long-term experiments on these cultures. Since they finish off after 5 to 10 divisions, they cannot be used for long-term studies. Now, primary cell cultures are considered by many researchers to be more physiologically similar to in vivo cells because they have not spent enough time in artificial condition. That is why they still resemble the original organ. The primary culture, we have two basic methods to obtain. First is explant culture. Small pieces of tissues are attached using plasma, clots or fibrogen to a glass or treated plastic culture vessel and then immersed in the culture medium. Then, after a few days, the individual cells will move from the tissue explant out into the culture vessel surface or substrate where they will begin to divide and grow. The second one is enzymatic dissociation. What do we use here? We use certain enzymes which are used to dissociate various cells from the organs. So it speeds up the process by adding digesting proteolytic enzymes. Why do we use proteolytic enzymes? Because the cells are bound by protein layers. So we have to separate the cells by dissolving the protein layer which is binding them together. So trypsin, collagenase are used to dissolve the layer. This creates a suspension of cells. These single cells are then placed in culture vessels which contain culture medium and then are grown. Now, Hayflick's phenomenon. This states cells will continue to grow and divide normally for a limited number of passages. When they get to a certain point, even if we give them proper nutrients, they stop dividing and they die. There appears to be a correlation between the maximum number of passages and aging. The number of passages decreases when the cells are harvested from the older cells. Obviously, the younger cells, they are available for more number of passages. Now, cell strains, the second type. Cell strains are the cells that have been adapted to culture. But unlike cell lines, they have a finite division potential. That is, they can divide up to a limited number of divisions, but more than the first one. Upon serial transfers of primary cells, a gradual selection may occur until a particular cell type becomes predominant. That is, when we start off with a mixture of cells, when we start growing a mixture of cells, a particular type of cell has more predominance finally. If these cells continue to grow at a constant rate over successive passages, these primary cells are referred to as cell strain. So, the ones which grow predominantly, they are known as cell strains. These cells have approximately 40 to 60 divisions as compared to 5 to 10 divisions in the earlier case. They are useful in production of vaccines. Now we have the third type, that is the cell lines. If the cells in a cell strain undergo a transformation process, that is spontaneous or induced changes in karyotype, morphology or growth properties, that makes them immortal. Immortal means they can divide indefinitely. as against the earlier two types where we had 5 to 10 divisions and 40 to 50 divisions. Here the cells divide indefinitely and now they are known as cell lines. It is not known how a diploid cell strain becomes a cell line, although the event may be mimicked by infection with oncogenic viruses. That is, it is said after infection with certain viruses, the cell is transformed and it becomes immortal. It doesn't die. It keeps on dividing and surviving in laboratory conditions. So cell lines often have abnormal chromosome numbers and may be tumorigenic when inoculated into susceptible animals. That is, when the cell gets transformed, it doesn't remain a normal cell anymore. It is transformed into a tumorous cell, a cancerous cell. Cell lines that have been derived from tumors often do not exhibit contact inhibition. Now, contact inhibition, what does it mean? In normal conditions, we grow up to a certain size limit and then we stop growing. That is due to contact inhibition of cells. When the cell volume reaches a particular volume, the cells stop dividing. Although they divide for wear and tear, but not for cell growth or the growth of the human being. But when the cells become tumorigenic, they lose this contact inhibition. They keep on growing, and that is the reason why the patients suffering from cancer, they have a lump developed in their body because of uncontrolled growth of the cells. Transformation of cells. Transformed infinite or established cells. They change from normal cells to cells with many of properties of cancer cells. These cell lines have actually been derived from tumors or are transformed spontaneously. So when we have to use cell lines in the laboratory, they can either be harvested from patient already having tumor or cancer, or we can take normal cells in the laboratory and transform them with the help of certain chemicals, mutagens, or viral infection. Chemical or gamma ray treated cells can become infinite with loss of growth factors. Viral infection with SV40T antigen can insert oncogenes and lead to gene alteration. No matter how transformation occurred, the result is a cell with altered functional, morphological and growth characteristics. That is, now we have a cancerous cell which can grow indefinitely in laboratory under artificial conditions. Cell culture systems. The cells may be loosely divided into two main types. The first is suspension cell culture, that is, they are anchorage independent cells. Now, what do we mean by anchorage? The cells which need a particular substratum to attach. Now, these cells, they do not need any substratum to attach themselves. They grow freely in the media. So, they are derived from cells which can divide and survive without being attached to a substrate. Examples, hematopoietic lineage cells. They can be maintained in the culture vessels and not tissue culture treated. This requires agitation for adequate gas exchange. This is easier to passage. Why is it easier to maintain? Because they are grown in a liquid medium known as broth in fermenter vessels. So fermenter vessels, to transport oxygen to all the cells, it has to be agitated. Agitated means it has to be stirred continuously, either with the help of baffle flask or by oxygen. The second system is adherent cell culture. This is anchorage dependent. Anchorage dependent means it requires a substratum to bind to attach to. So it must adhere to a surface or to survive. It forms monolayers. Cells derived from different tissues like liver, breast, they are the ones which are adherent cells. Growth is limited by surface area. It will cease proliferating once they become confluent, completely cover the surface of the particular apparatus or the container in which they are growing. Cells are dissociated enzymatically or mechanically from the surface. Now, what is the growth cycle that is followed here? Growth cycle in attachment culture. Eukaryotic cells in attachment culture, they have a characteristic growth cycle which is similar to bacteria. The growth cycle is divided into three phases: lag phase, log phase, and the stationary phase. And then comes finally, there is a decline phase. So we first discuss the lag phase. Lag phase. This is the time following subculture and receding during which there is little evidence of an increase in cell number. Why is it so? When we pull out certain cells from their original places, like organs, then since the cells are already acclimatized to surviving in their environment, and when we take them out from their original environment and put them in artificial environment in laboratory, then they take some time to adjust to the new conditions, the newly provided conditions, the artificial nutrients that are provided in the laboratory. Cells take some time to adjust, and during that time they do not divide and grow. They just try to maintain themselves. It is a period of adaptation during which the cell replaces elements lost during trypsinization. Trypsinization means treating with enzyme trypsin, attaches to the cell surface and spreads out. Second phase is log phase. This is a period of exponential growth. Since the cell has adjusted itself in the new medium, now it starts dividing rapidly to utilize the nutrients as well as the newly found space, to fill up the space completely. So, this is the period of exponential increase in cell number. The length of log phase depends on the seeding density, the growth rate of the cells. It is the optimal time for sampling since the population is at its most uniform and viability is high. Sampling as well as for any kind of research purposes, the scientists generally prefer taking cells from the log phase. Since it is the fastest growing, it is the most healthy phase, that is why these stage cells are preferred for any kind of research work. The third phase is plateau phase. Toward the end of the log phase, the culture becomes confluent. All the available growth surface is occupied and all the cells are in contact with the surrounding cells. Plus, all the nutrients that was given, that were provided in the beginning starts becoming exhausted. And waste materials starts to gather up. Waste material starts accumulating. And these are the reasons, since nutrients are getting over, wastes are getting accumulated, the cells, they stop or they slow down. They slow down, their division slows down. Following confluence, the growth rate of the culture is reduced and in some cases, cell proliferation ceases almost completely. Now, it is a stationary or a plateau phase. This is the graph depicting all the three phases in a cell cycle. We have a lag phase initially, approximately for 2 days, and then from third day onwards, the cells start picking up their division. They go on dividing till 6th day continuously, and after 6th day, they start the plateau phase. What is the morphology of cells? Cultured cells are usually described based on their morphology, shape and appearance. Now there are two basic morphologies. Epithelial-like, that is, cells that appear flattened and polygonal in shape. The second is fibroblast-like, cells that appear thin and elongated. The culture conditions play an important role in determining shape, and that many cell cultures are capable of exhibiting multiple morphologies. What is the basic requirements for successful cell culture? The first necessity is a well-established and properly equipped cell culture facility. Class, we need a proper, sterilized, well-equipped laboratory where cell culture can be performed. We cannot perform cell culture in any open laboratory. The laboratory has to be completely sealed to avoid outside contamination. So, all the facilities should be equipped with the following. First is a certified biological safety cabinet. It is also known as laminar air flow hood, in short LAF. This protects both the cells in culture and the worker from biological contaminants, that is the scientist as well as the cell. They are prevented, they are protected from each other. They are protected from infection traveling from the cell to the scientist or from the scientist to the cell. Second, we need a centrifuge, which is preferably capable of refrigeration. Why? When we centrifuge at high speeds, friction is bound to generate some heat. So to avoid heat, we need refrigerated centrifuges. Then we need a microscope for examination of cell cultures and for counting the cells. Then we need a humidified incubator set at 37 degrees C with 5% CO2 in air. This is specially required for animal cell cultures. CO2 presence, because pure oxygen cannot be used for growth of animal cell cultures. At 37 degrees C, water bath filled with water containing inhibitors of bacterial and fungal growth. Obviously, animal cells can be outgrown by bacterial and fungal infection. Since bacteria and fungi, they divide faster as compared to animal cells. So we need to have certain inhibitors to stop the growth of bacteria and viruses and fungi. Next, the second requirement for successful cell culture is the practice of sterile technique. Prior to beginning any work, biological safety cabinet, it should be turned on and approximately run for 15 minutes to purge the contaminated air, to remove the contaminated air which has been there. Then, all work surfaces within the cabinet should be decontaminated with an appropriate solution. We here use 70% ethanol or isopropanol for decontaminating the surface. We take it on cotton and just wipe out the surfaces to decontaminate or sterilize them. Any materials required for the procedure should be similarly decontaminated, like the glass stirrer, like the forceps, the scalpel, everything has to be sterilized. This is especially important if solutions have been warmed in a water bath prior to use. The workers should put on appropriate personal protective equipment for the cell type in question. Depends what kind of cell type we are using. If the cell line is pathogenic, we have to be extra careful. Gloved hands should be sprayed with decontaminant prior to putting them into the cabinet, and the gloves should be changed regularly if something outside the cabinet is touched. Once we take our hands out of the laminar airflow hood, we have to change our gloves because the outside atmosphere is not sterilized. Care should be taken to ensure that anything coming in contact with the cells of interest or the reagents needed to culture and passage them is sterile. It has to be autoclaved to make them sterile. Or maybe if it is heat labile, then it has to be filter sterilized. The third necessity for successful cell culture is appropriate quality controlled reagents and supplies. There are numerous suppliers of tissue culture media and supplements. Examples are Invitrogen, Sigma, BIOTECH, stem cell technologies, etc. Similarly, there are numerous suppliers of plasticware needed for most culture applications. Like culture dishes are required, flasks, tubes, disposable pipettes, etc. Then, the fourth necessity is knowledge and practice of fundamental techniques. The scientist working on cell culture should be knowledgeable, should have practiced earlier, involved in the growth of cell type of interest. Majority of cell culture carried out by investigators involves the use of various non-adherent or adherent continuously growing cell lines. Now these cell lines can be obtained from reputed suppliers. It's very difficult for us to prepare a new cell line. We have available already established cell lines with manufacturers. So we can just order them as per our needs. The American Tissue Type Collection, that is ATCC, or DSMZ, the German collection of microorganisms and cell cultures. Alternatively, they can be obtained from collaborators. Regardless of the source of cells, it is advisable to verify and identify the cell line to ensure that it is free of mycoplasma contamination. The moment we get the cell lines from the manufacturer, we need to identify, we need to see whether there is any contamination or not. Might be during transport there were contaminations which entered the package. Now cell culture medium, the medium in which the cell is grown. What all are required in cell culture medium? Cells have complex nutritional requirements that must be met to permit their propagation in vitro. Different types of cells have different growth requirements. So as per the cell type we are trying to culture, we have to modify the media composition. So number of chemically defined formulations are available and have been developed that support the growth of variety of established cell lines. Although Some serum-free media are available and some cell lines have been adapted to growing in such a medium. Most of the cell lines require addition of approximately 5 to 10% serum as a supplement so that cellular multiplication is promoted. Fetal bovine serum is the best one. The various nutrients which are required in a medium are glucose, fats and fatty acids, lipids, phospholipids, sulfolipids, ATP, amino acids, vitamins, minerals. Serum, it can provide various growth factors, hormones and other factors needed by most mammalian cells for the long-term growth and metabolism. L-glutamine, it is an essential amino acid which is required by virtually all mammalian cells grown in the culture. It is used for protein production as an energy source and nucleic acid metabolism, that is formation of new nucleic acid. It is also labile in liquid cell culture medium than other amino acids. The rate and extent of L-glutamine degradation are related to storage temperatures, age of product and pH. Buffering in the cell culture. Due to metabolism which is taking place in the cells which we culture in the laboratory, they might secrete acid or alkali, and accordingly the pH of the medium may turn acidic or alkaline. But depending on the type of culture we are making in the laboratory, the pH has to be maintained at a particular point. That is why we need buffering in cell culture. So a pH indicator might be included in the original formulation to permit direct observation of pH. Optimum pH should be between 7.2 to 7.4 for the animal cells. That is animal or mammalian cells. So we use bicarbonate buffer system for the maintenance of the pH. Buffering, generally in the cell culture medium, pH indicator phenol red is used to analyze the pH of the environment in which the cells are growing. How are we going to know whether the pH is turning towards acidic or alkaline? We need an indicator which can turn a particular color. That is, it can change color depending on the change in pH. So here we use phenol red. Phenol red is yellow in acidic medium, that is around 6.8, turns tomato red at neutral pH, that is 7, red at alkaline pH, that is 7.4, and blue at increased basicity, 7.6, and then finally purple at high pH. So looking at the color of the medium, we can judge what is the pH, what is the range of pH right now. If it is within the allowed range, fine. Otherwise we'll have to put in acid or alkali depending on what kind of adjustments the medium requires. What are the supplements to the medium? The first supplement that we add to the medium is antibiotic. Why do we need to add antibiotics? Because to prevent the growth of unwanted cells. We just want the mammalian cells to grow in our medium. We do not want the bacterial, viral, fungal cells to grow in the medium. So we need to add antibiotics. So prevention of contamination by different microorganisms, bacteria, mycoplasma, fungi is the most important part of all animal cell culture. The risk of contamination during culture can be avoided by adding different antibiotics such as penicillin, streptomycin, gentamicin, nystatin. The routine use of antibiotics is generally not recommended because it may lead to relaxation of aseptic technique. Resistant microorganisms may develop and they may contaminate our mammalian cell culture. Microbial growth may be controlled, but biochemical alteration may be produced in the mammalian cells which we are trying to grow and work upon. Now, what is the temperature and humidity that needs to be maintained? Optimum temperature is required and it is approximately 37 degrees C for the mammalian cells. What is the humidity? For cell growth, 100% humidity is essential. We do not want our cells to dry down and shrink. So, 100% humidity is required so that there is no evaporation. Now, how do we store the medium? Once we prepare the medium, the cell culture medium has to be properly stored. For long-term storage, it should be frozen. And on a short-term basis, medium should be kept at 4 degrees C and warmed at 37 only at the time of performing of the experiment. Now the culture vessels. The vessels which we use for culturing of mammalian cells, they are very important and they have to be sterile and of good quality. Culture vessels provide a contamination barrier to protect the cultures from the external environment while maintaining proper internal environment. For anchorage dependent cells, the vessels provide a suitable and consistent surface for cell attachment. Other characteristics of vessels include easy access to cultures and optically clear viewing surfaces. What do you mean by this? The culture vessel should have an opening from where we can either add the cells or extract, that is harvest the cells. Plus, we should have clear viewing surfaces. What is going on inside the vessel? We need to monitor it continuously. So we should have something so that a clear view is available. We can view what is going on, how the cells are growing. We can at least see what is the morphology of the cells which is growing inside the vessel. It is very important because once the cells turn into transformed cells, they turn into tumorous cells, cancerous cells, their morphology changes. So depending on our requirement, we want the cells to grow in particular manner. So the first culture vessel which we are going to discuss here is flask. Plastic flasks, they are available with a range of growing areas, a variety of shapes with several different neck designs. As you can see here, we have a flat flask which can be laid down horizontally for the growth of mammalian cells. The flask surfaces are specially treated for growing the anchorage dependent cells. These flasks are mainly for growing anchorage dependent cells. Suspension cells are not grown in these flasks. So the description, as you can see, is given here. We have different types of flask and their growth area and the volume in which they can work and what kind of yield they give. So depending on our requirement, whether we want to work with a small volume, medium volume or large volume, we can choose the respective flask. The second vessel is cell culture dishes. You can also call them Petri dishes. Petri dishes are available in circular form and they have a cover so that sterile conditions can be maintained. They can be covered so that any contamination does not enter easily. Also, Petri plates can be sealed with the help of a tape. Cell culture dishes offer the best economy and access to the growth surface. The cell culture dishes surfaces are specially treated for growing anchorage dependent cells. Again, here also, what do? We use a media, that is a nutrient media. Nutrient media supplies all the nutrients to the growing cells. Plus, here we add agar. Agar is a gelling agent, a solidifying agent. So, the gel, it forms with the nutrients, it forms a semi-solid material which is in the Petri, and on the surface of this material, the solidified, the semi-solid material, we can grow the microorganisms or mammalian cells. So, cell culture dishes offer the best economy and access to the growth surface. The cell culture dishes surfaces are specially treated for growing anchorage dependent cells. Again, we can see we have different sizes available in the Petris, and these offer different growth area and the volume and the yield. So depending on our requirement, we can choose a particular size. Third is multi-well plates. These multi-well plates are actually having small wells, number of wells in them. They can be of different sizes depending on requirement. They can have approximately 6 wells, 10 wells, 20 wells, 100 wells, depends what amount we want to work with. So multiple plates, they offer significant savings in space, media and reagents when compared to an equal number of dishes. Since there are multiple wells within the same plate, so multiple tests can be performed within a single plate, and this helps us to save on time, space, media, reagents. Now, surface coatings. Most tissue culture work uses disposable polystyrene vessels. The vessel surface is then treated to render it hydrophilic. Most cell lines are cultivated on treated plastic surfaces in dishes or flasks. Some fastidious cell lines require further treatment of growth surface before they will attach and proliferate. The most common techniques include coating the surface with serum, collagen, laminin, gelatin, poly-L-lysine or fibronectin. So students, here we finish with our today's topic, that is cell culture, and we'll continue it tomorrow. Thank you.